Hello. Hi, my name is Harris Obiyama, and I am an undergraduate student of Sam Houston State University, and I am presenting my experiment to the URS, the Undergraduate Research Symposium. Now, my experiment is talking about whether mitochondriopathies can be rescued by manipulating Hippo pathway signaling. So starting out, the Hippo pathway is a vital regulator of organ size in a Drosophila melanogaster fruit fly in humans. With Yorkie results in an increase in cell survival, cell proliferation, and glycolysis. And Yorkie is like a specific component part of the Hippo pathway. And as you can see in this diagram, there are homologs of the Drosophila and the humans as they relate. So the background on mitochondriopathies or mitochondrial defects, every 30 minutes, a child is born who will develop a mitochondrial disease. And mitochondrial defects is the common metabolic disorder. And also in mitochondrial defects, there's a decrease in cellular energy and it leads to cell death or cellular apoptosis. And also it can lead down the path to improper tissue growth. So Drosophila melanogaster, we use it and why is the perfect model organism? So it's one of the cheaper model organisms we use, and also 75% of the known human disease genes have a recognizable corresponding match to the fruit fly genome. Also, it can be easily replicated, and it's easier to have genetic manipulation, and also there's a shorter lifespan, meaning rapid progress. Now, for importance, this is important with the ATP synthase, which is part of complex 5 in the electron transport chain, and it's a protein complex that actively transports ions, specifically protons, in and out of the mitochondria, and it helps with tissue growth and then just overall cellular activity. So how do we induce this? So our model for the mitochondrial defect is decreased ATP synthase function, meaning decreased in energy production. So to do this, we use RNAi against the subunits of complex 5. And there are many subunits, which is alpha, beta, and gamma, but we were primarily focusing on alpha, which is also named as bellwether. And with this being said, bellwether causes the decreased viability in wing pouch size in the fruit fly overall. Now, generally, RNAi is a tool used by researchers to interfere with the messenger RNA and impeding the target protein's production. So it's a very useful tool. So our main hypothesis is with Yorkie, which is the Hippo pathway effector that we briefly went over, that Yorkie activation can rescue poor growth or poor survival of cells with the mitochondrial defect. Now, before we continue, I need to go over our experimental approach of the main tool we use, which is the Gal4-UAS system. So the Gal4-UAS system is a transcription factor activator within yeast. That's Gal4. And with that being said, with Gal4, it activates transcription of what we want expressed by attaching to the UAS sequence. Yes. And then with that being said, it expressed what we're looking for, or what we're our prime focus of what we want expressed. So if, for example, what we're expressing X, it can be bellwether RNAi, it could be Hippo RNAi, it could be luciferase or Hippo overexpression, or an overexpression of Yorkie, which is Yorkie S1278 or Yorkie 1SA. Now, with this being said, we're going to see the expression for the experiment. We're going to see the expression of these. We're going to see these expressions in the wing pouch of the developing wing disc of the Drosophila melangasters. Okay. Now, there were two approaches. So the first approach is to inhibit the Hippo pathway using Hippo RNAi. Remember, RNAi was the tool to impede the RNA machinery. So is it leading to the Yorkie activation? The second approach is using UAS Yorkie S1278. Right, we are overexpressing that transcription coactivator to see whether it can rescue those degenerative cells. Okay. Now, as you can see, we go from our control. Our control is nubbin with luciferase. You can see it has a pretty normal, with this sign, it looks like a pretty normal growth compared to its competitors with nub Hippo RNAi, which has slightly more growth versus the luciferase and nubbin. And then the nub, you can want to say, which is the overexpression of nubbin, has, you can see, is by far more of a more cellular proliferation, more growth seen with from what we've known. So we decided to use that information we already knew to attach or to combine with our induced mitochondrial defect or the ATP synthase. So we were hoping what could occur. So with this being said, these were the results from the experiments. As you can see, nub-gal4 luciferase has its own, this is expressed in the wing pouch within the overall disc. That's the control. And compared to the rest, as you can see, nub-Yorkie, when I say, which is the overexpression of Yorkie, the overexpression of Yorkie, you see it's 5 times to 6 times more growth of the wing pouch versus the overall wing disc, right? So in terms of, and yeah, so with that being said, with the other competitor of nub Hippo RNAi with luciferase, you can see the wing pouch is bigger and the disc is slightly bigger, but it's not the same as our control of nub-gal4 and luciferase. Now we conjectured this with nub-gal4 and bellwether, and then you can see that the overall wing disc, the nub-gal4 is the more similar one out of the rest, and the wing pouch is similar, but it's still bigger by a small amount. But then the crazy part is with the overexpression of Yorkie, when I say of Yorkie, which is Yorkie when I say, with the ATP synthase with the mitochondrial defect caused compared to the control, it caused like a more of a titration or a smaller wing pouch to the nub you're going to say to luciferase. So it controlled its own action. But at the same time, compared to our control, when you compare this nub you're going to say with bellwether to its control, you can see that the wing pouch is still a huge amount. Not as much as the control of nub you're going to say, but it's still a huge amount compared to our nub-gal4 luciferase. And still more experimentation is needed to be done for nub Hippo RNAi with bellwether. So this is the qualification of our results. So Yorkie activation rescues growth of bellwether RNAs and RNA cells. So we measured 5 to 10 wing discs for pouch area and total area and calculated the ratio. We then plotted that on this graph right here, as you can see. And then when that happened, we performed the Welch and Nova test and found that they were significantly different. And the pair t-test revealed that the mean difference between nub, you're going to say, and bellwether and nub luciferase is 0.5 and was significantly different with a p-value of 0.0001, right? And the nub you're going to say plus luciferase is significantly different than the nub luciferase. And what you hear, this is our control, which is the nub luciferase. That's where we're trying to get all of our other competitors or other variables to be and try to simulate that with these different competitors. So as you can see, the nub you're going to say luciferase is a high extreme compared to the control, and then which means that it by itself is really itself proliferates a high amount, and then with bellwether it decreased that amount, but it's still pretty high, and its data is pretty awesome and astounding. And yeah, so experimental conclusions, we can see that the genotype, I mean, with the data that nubbin and luciferase, nubbin and the ATP synthase still compared to the control is slightly more of a cell proliferation than original, and then the nub you're going to say is still a huge amount of cell proliferation because it kind of overshot the goal what we were intending it to, and the nub Hippo RNAi still needs to be disclosed. So the take-home message: so the defect in the complex is one of the most common way to get a mitochondrial defect. So, and which we were trying to find a way to fix this through the Hippo pathway. So with Yorkie when it say the too much cellular proliferation as seen with overexpressed Yorkie with the ATP synthesis can be cancerous in the mammalian system. But as we saw in it's still a progress. It's still progress. And also ATP synthesis causes overgrowth, but not as overexpressed with Yorkie by itself. So the model has been somewhat been saved with Yorkie, but it's still overshot the goal. So going forward, we should be able to determine if we can reduce or possibly remove the possible mitochondrial defects found in most of these diseases, possibly. And to go through that, we need to find a weaker Yorkie activation because that was highly activated Yorkie. And then we need to observe whether this can affect the other different types of cell types. The cell types we primarily looked at was epithelial cell types. We could look at neuron cells, muscle cells, and maybe adipocytes. And we need to try to assess the delay in development because with bellwether, there's sometimes a development delay with the larvae that comes out of the specific genotype. But also how we're able to tune or manipulate these two factors of the ATP synthase and the York in a way that goes against the deficiency in the mitochondria. And more experiments and data are needed. So yeah, I just want to acknowledge this. These are my coworkers and my Professor Dr. Atkins. Thank you so much.
You're welcome.